Hello to my friends joining us via recording. We are wrapping up our discussion of the special senses, lesson number 13. We have talked about the process of vision in the video that says it's part 1 for lesson 13. We talked about the processes of hearing and equilibrium or balance in part 2. And we also started to talk about taste and smell in part 2. We're going to wrap up the sense of taste in our time tonight, and then maybe do a little bit of reviewing for some of our special senses, maybe some of our spinal cord stuff, the things that are going to show up on our last unit exam. So one of the things that we did not talk about this morning in our session together about special senses were the specific type of papillae that we find on the tongue. When you look at your tongue and you see bumps on it, what you're seeing are things called papillae. Now, papillae are not the same thing as taste buds. Taste buds, like I see right here, are very small, and they're actually inside of those bumps that you see. So most of your tongue is covered in what we call fungiform papillae. And they get their name Fungiform Papillae because if you look at them from the side, they kind of look like a mushroom or fungus. This is the most common type of papilla that we have on the tongue, or bump on the tongue. These have a moderate number of taste buds inside of them. If we want to see the type of bumps that have a ton of taste buds, we actually have to go to the back of the tongue. So the back of the tongue, where we start getting into the throat that we see back here, the back of the tongue has what's called Valate Papillae, and Valate Papillae are arranged in the letter V that I see right here. So we don't have a lot of these Valate Papillae at the back of the tongue, but they have a ton of taste buds inside of them. So in the process of swallowing, you actually detect more taste information at that point. These special kind of bumps that we see here on the side of the tongue are called Foliate Papillae. For my friends who've had a chance to work through the lesson, do you remember what's special about these Foliate Papillae? What's special about these ones? Do you remember? Yeah, so Foliate Papillae are only found in children. There's something that you grow out of as you grow older. So children actually have extra sets of taste buds on the sides of their tongue arranged in these Foliate Papillae. So when we say that kids just don't like vegetables, aside from just being stubborn, maybe those vegetables actually do legitimately taste different. At least that's what I tell myself when I can't get my daughter to eat vegetables, right? It must just be those Foliate Papillae. She's tasting something different. Vanessa asked when they grow out of them. That's a good question. I don't know for sure. I'll have to look into that a little bit more. I suspect it would be maybe in adolescence, maybe sometime around puberty, but I can't say for sure. I know that they're definitely in young children and probably like elementary age children, but I'm not sure when they go away. I just know that we all can't blame our Foliate Papillae since we don't have them anymore. Remind me, by the way, from this morning, we said that the process of gustation, which is the fancy word for taste, we said that this was a chemical sense. What kind of information does it tell me when I say that the process of tasting or gustation is a chemical sense? What are some of the things we talked about this morning that means? It's a chemical sense. Perfect. Yeah, so first thing it means when it's a chemical sense is that what we're actually tasting, we taste chemicals. And remember from this morning that we said the special name of those chemicals, they're called tastants. Tastants. So the first thing it means when I tell you it's a chemical sense is that what we actually are detecting are chemicals. Second thing that it means is it tells me the kind of receptors that I use to detect things. So if it's a chemical sense, it means I'm going to use chemically gated channels to detect this kind of sensation. This is some great review from unit number 1 back in the day. If I'm a chemically gated channel, what opens me? Chemically gated channel. Not a trick question, right? If only all the questions were this easy. Yeah, so when we were talking about neurons, the chemical that opened those gates was neurotransmitters. When we're talking about chemically gated channels on a hair cell, a gustatory hair cell, what opens those are the chemicals in your food. So chemically gated, typically when we're talking about a neuron, that's going to be neurotransmitters. But this name in general just means that some kind of chemical opens it. So chemically gated channels on gustatory hair cells, those are going to open with the chemicals in your food. Chemically gated channels on a neuron are going to open with neurotransmitters. It's all about a chemical opening the gate, though. There's one more thing. I don't think I saw it in my notes here. One more thing that I know, if it's a chemical sense, what do I have to do to these chemicals to be able to detect them? What has to happen to them for me to be able to detect them? Yeah, they have to be dissolved. Chemicals must be dissolved. Meaning I've got to put them in something, in a solution. The particular solution that I put my tastants in is everyone's favorite, saliva. Actually, that's better than what we use for smell, right? What's my solution that I use when you're smelling? We don't use saliva to help us smell. Yeah, we use mucus to help us smell. But it's the same idea. We've got to dissolve the chemical that we're detecting. When I dissolve it, then that chemical can go and find chemically gated channels that open up on my gustatory hair cells for the sense of taste, or my olfactory hair cells for the sense of smell. So it's a chemical sense. We are tasting chemicals called tastants. We taste them because they open chemically gated channels, and we have to dissolve them to be able to detect them. Yeah, so Nicole had asked the question about what exactly we're looking at here. We are correct. This is a taste bud. So one taste bud is made out of a bunch of gustatory hair cells, but all of the bumps on your tongue have more than one taste bud in them. So let me do a really terrible drawing here. We're going to draw a little fungiform papilla. These are the ones that are the most numerous on your tongue. We have a bunch of these papilla on the tongue. Inside these mushroom-shaped bumps, you find little taste buds. So I'm doing a terrible job of drawing for you the locations of taste buds. So a single papilla, a single bump that you would see on the surface of your tongue, inside it in various places, you're going to see these little clusters of cells that look like this. So on top of each of these clusters of cells is the opening, the taste pore that you see labeled here. There'd be a little taste pore opening on each of these places for that saliva to go down inside of with the chemicals inside of it to activate those cells. So the surface of your tongue has bumps, and those bumps maybe have 5 to 7 taste buds in them if they're a fungiform papillae. If they're one of these Valate Papillae along the back, they're much, much bigger, and they can have like 50 to 100 taste buds. But each taste bud has this same structure that we see here with little hair cells inside of it that are activated, that their gustatory hairs are activated by chemicals. By the way, these gustatory hair cells, they actually turn over pretty rapidly, meaning we replace them pretty rapidly. We are grateful for that because if you ever drink coffee that's too hot or take a bite of your food before it's cooled down, you can actually damage or even kill the gustatory hair cells. So those cells replace themselves pretty rapidly. I can't remember, though. In the guided lesson, Did I mention for you when we slow down that process of replacing them? Did I put that in the guided lesson? Crickets are making me think I probably didn't. Yeah, so in the elderly population, this process of division of the gustatory hair cells, of making new ones, it actually slows way down. So with aging, your sense of taste declines dramatically, which is why in older individuals, the interest in food declines, because the taste sensations that would come from food, when we start losing these gustatory hair cells, when we stop making new taste receptors, food is not nearly as enjoyable. So as a person ages, their sense of taste declines. The other thing that is cool with the sense of taste, sometimes you might say that your senses changed, like you used to like something and now you can't stand it. Because we're constantly replacing these gustatory cells, our cells that detect things, what they taste like, it is legitimately true that something may have literally tasted different in the past, and now it tastes better or it tastes worse because we have new gustatory hair cells that have grown into our taste buds. So just some little fun trivia about taste buds. If it wasn't in the guided lesson, don't worry about it. I'm not going to ask you about it, but that is part of why the sense of taste declines, because we stop replacing these cells here. Were there any other questions about the process of tasting or smelling, my two chemical senses? Nicole asked, can all the taste buds detect all 5 taste sensations? I believe the answer to that is yes. Detecting the different taste sensations comes down to which kinds of gustatory hair cells are inside of the taste bud. So assuming that we have a variety of different kinds of hair cells in the taste bud, we should be able to taste all the various sensations at different places on the tongue. There used to be if you do a Google search, you can find kind of a debunked theory or a debunked map of where you taste different sensations on the tongue. It used to be thought that there were certain places on the tongue where you tasted sour and certain places where you tasted sweet. It came out years after that study that they tried to replicate it many times and failed miserably. It really comes down to almost like test subject bias. It's really hard for you to figure out where on your tongue you're tasting something because you have the ability to taste it anywhere. So as long as we have the right kind of hair cells everywhere, you can taste every sensation everywhere. The way that you figure out exactly what you're tasting is by doing our favorite neuron word, integration. Integration. When we're talking about tasting, we're integrating sweet and sour, salty, umami, my personal favorite. I'm missing one. Sweet, sour, umami. I'm missing 2. What am I missing? Why is my mind going blank? Bitter. Yes, thank you. My favorite. How could I miss out on bitter? Why do I love bitter so much? What's bitter found in? Do we remember? Yeah, bitter is found in coffee, my personal favorite. So bitter, favorite taste sensation. What else are we missing? Bitter, sweet, sour, umami. Salty. Okay, I said salty already. Maybe, I don't know. So the 5 taste sensations, it's a combination of, oh, I'm tasting some salty, I'm tasting some sweet, and some sour all at once. That helps me to figure out what it is that I'm tasting. Or you taste that steak, and you can taste the salt on the outside to season it. You're putting together the umami and the salty, and it helps you to taste a really good steak. The process of tasting things, we integrate all of the kinds of information that we receive. Nicole asked about the Valate Papillae. For the sake of our class, yes, we're going to say that the Valate Papillae is just this V line right here. These are different bumps that are in the throat. I don't believe they're technically called Valate Papillae because they're not on the tongue. But these bumps are filled with taste buds as well. So you can actually do the process of taste in your throat, at the top part of your throat. Not with Valate Papillae, because that papillae word specifically means bumps on the tongue. So this is the Valate Papillae. These are some of the bumps that have taste buds in the throat. I think I'd seen a question that somebody else answered too. Yeah, so taste buds versus papillae. Papillae just is a fancy word for bumps, and inside of those bumps are the little taste buds that we use to taste things. How do we feel about taste and smell? Okay, here we go. Why don't you all send me an emoji of a food that either you are eating or you wish you were eating. Let's do that. I'm not currently having coffee. It's a little too late for that. Christina's having coffee. Believe me, Christina, I know that life. Ooh, pretzels. That would be delicious. I'm feeling some pizza. Yeah, Gloria, it would be good for exam 4 to be familiar with, especially, I don't think there's too many ions that I ask you to know for the taste sensations. But for example, we want to know that sour is caused by those hydrogen ions. Salty is sodium and chloride. So there's just a few that you'd need to know it for. Do go ahead and know them. Ooh, lollipop. I like it. Hamburger. We got some. Is that ramen? Is that what that's a bowl of, maybe? Hannah's like, give me all the things. I like it. Jacqueline is channeling her inner cat and wanting to eat some fish. This morning we were talking all about the cats. So anyone who didn't make it here this morning when we were talking about, although I guess that was probably group work, wasn't it? When we were talking about hyperopia versus myopia. So alas. Okay, what I will do then, since we seem to be feeling okay about smell and taste, I want to build a compare and contrast for us, because you know how Dr. Allis loves her compare and contrast. We are going to do a compare and contrast of equilibrium types. So when we talk about the types of equilibrium, we talked about them this morning. We talked first about what's called static equilibrium, and we talked second about what's called dynamic or rotational equilibrium. When we talk about these two types of equilibrium, let me move it to have a little more space. When we talk about these types of equilibrium, there are several things that we need to know about them. So let's make a list and then we'll fill in our table together. First thing we need to know about these two types of equilibrium is what are they detecting? In other words, if you're out of balance with one of these types of equilibrium, what exactly does that mean? Which way are you bending, or which way are you turning? That kind of thing. What are they detecting? That's the first thing we should know. The second thing we should know about these equilibrium is where in the ear we detect these kinds of sensations. So there's different parts of the ear that detect these things. We need to know that. The next thing we should know with these are the major structures that are involved in detecting each of these types of equilibrium. Because we talked about today, there's some specific words that we use to describe what we detect with these things. So let's start with these three main questions, and we'll fill it in here for static equilibrium and dynamic equilibrium. When I'm talking about the way that you detect whether or not you're turning or if you're spinning, which kind of equilibrium helps me to know if my body is turning or if it's spinning? That's either way. Yeah, that's going to be my dynamic equilibrium. When we talk about body spinning or rotating, its name is rotational equilibrium, right? So when your body is spinning or rotating, that's what I detect in the process of dynamic equilibrium. When I talk about static equilibrium, how could we describe what I'm detecting? What kind of movements might you say static equilibrium helps you to detect? I like it. Head flopping. Head flopping. We could say, like I mentioned this morning, I kind of like to give the joke, the ways you fall asleep in class, right? So with static equilibrium, it's relative to gravity. Relative to gravity. Let's put that up there. Gravity wants to either pull your head forward or backwards. So flopping that head forward or backwards. Static equilibrium is body going forward or backward. Relative to gravity. Gravity just wants to pull you forward or backward. So Nicole asked a good question. If this is just the head or if it's the entire body. Mostly when we talk about the kind of information that's going through the vestibulocochlear nerve related to balance, the vestibulocochlear nerve in particular is listening to what's going on with your head. But what's going on with your head is kind of the same thing that's happening with your body for the most part. The body has another kind of receptors too, though, that help us know if we're bending forward or backward. Here's a word, I think it's probably popped up at some point in the class, but we didn't talk a lot about it. We have a type of cells called proprioceptors. They detect a particular kind of information. Did we ever talk about proprioceptors or proprioception in our class? We may not have used the technical word for it. Okay, so we have talked about it. I just didn't use that technical word on you. Proprioceptors are receptors that detect stretching. I know we talked about stretching because we said that was a general sense. So in general, if you're trying to figure out if your whole body is going forward or backwards, if your whole body is rotating, to know what's going on with your body, we use information from our proprioceptors, that's the stretch detectors, for what movements we're doing. We'll couple that with the information that we have about static equilibrium or dynamic equilibrium to figure out if we're falling forward, if we're tripping forward. So we couple stretch information with balance information most of the time. So if we want to get super technical, when you look at your pictures in the notes and it only shows you the head, either when we're talking about going forward or backwards or when we're talking about spinning in circles, recognize that probably the whole body is doing it too. It's just the way we figure out that our whole body is doing it is through those proprioceptors, those stretch detectors. So I don't care however you memorize it. I'm not going to get that nitty gritty on the test and ask you, is this something that it only detects for the head or for the whole body? What we want to know for static equilibrium, forward or backward. What we want to know for dynamic equilibrium, spinning around, we're rotating. When we talk about detecting forward and backwards, static equilibrium, there were two places in the ear, two areas that do static equilibrium. What was the name of those places that do static equilibrium? Forward and backward. Yeah, the forward and backward places in the ear are called the saccule and the utricle. These were the two really big fluid-filled areas right next door to the cochlea, the saccule and the utricle. They're going to help us knowing forward versus backwards. So Nicole asked a question, and we didn't technically discuss it this morning, but yeah, we're using our powers of inference here. She asked what kind of fluid fills the saccule and the utricle. The fluid that we have in the saccule and the utricle helps to activate the hair cells that live there in the saccule and the utricle. And remember that the type of fluid that activates hair cells is called endolymph fluid. So I have endolymph fluid that fills up the saccule and the utricle. Remind me again, why, what's going on with the chemistry or the recipe for endolymph fluid? It has a lot of something. What does endolymph have a lot of? Lots of, exactly, potassium. Lots of potassium. So static equilibrium, detecting whether we're going forward or backwards. That's done by the two big areas called the saccule and the utricle. They are filled with a kind of fluid called endolymph fluid. When we talk about dynamic equilibrium, we don't use the saccule and the utricle. Where or which parts in the ear do dynamic equilibrium? Who's that? Yeah, Gloria's right, Christina's right. The place in the ear where we do dynamic equilibrium are the places called the semicircular canals. The semicircular canals. And remember that you actually have 3 of these canals. Each of these canals is detecting the precise rotational movements that your bodies are doing. Because keep in mind that we don't just rotate our head when we're holding it straight up. Sometimes we rotate our head when it's at an angle. Sometimes we rotate our head when it's at the other angle. For us to know precisely how we're rotating, we actually have 3 different semicircular canals that help us to detect the exact movement that we're doing. Which kind of fluid do I find in the semicircular canals, by the way? What fluid's inside there? The semicircular canals. Any guesses? Yep, we're back to that endolymph. Endolymph. When in doubt, think about if we're talking about a part of the ear where we're actually hearing, or the part of the ear where we're actually detecting equilibrium. If we're in the places that actually detect things, we're going to have endolymph fluid. Because remember, this morning when we talked about the cochlea, the place where you do hearing, there were 3 tubes. 2 of those tubes we didn't actually hear the sound waves in. 2 of those tubes just transported the sound waves. But what was the name of the tube in the cochlea where you actually hear sound? Which tube in the cochlea helps us to actually hear sound? Yeah, the tube in the cochlea where we actually hear sound is called the cochlear duct. The cochlear duct. So that's where we, I'll say, perceive sounds, where we perceive the sounds. Because that's where we perceive them or where we hear them, that is the part that is also full of endolymph fluid. So I have endolymph fluid in the cochlear duct where I hear sounds, and I have endolymph fluid in the semicircular canals that help me with spinning, and I have endolymph fluid in the saccule and the utricle that help me with knowing if my head is going forward or backwards. I have a couple other tubes in the ear. I'm not going to make you spell them because they're terrible. Let me type them for us. Scala vestibuli and scala tympani. Those places don't actually hear sound. What's the name of the fluid that fills these places, that just moves my sound waves for them? I don't actually hear it. Yeah, those tubes are filled with what's called perilymph fluid. Perilymph fluid just transports the sound waves. I don't actually hear when I've got sound waves in perilymph. When it's in endolymph, I can hear it because it activates my hair cells. Or when we're talking about rotation, when we're in that, when we open up our channels for the endolymph, that's going to allow us to detect movements. So now we're to the point where we're comparing the structures that are involved in static equilibrium and dynamic equilibrium. Let's start with one structure that I have in both of these kinds of equilibrium. Can anyone think of the name of something that I have in both of these kinds of equilibrium? Yeah, Nicole's right. So something that I use in both kinds of equilibrium, my cells that actually do the detecting for both static equilibrium and dynamic equilibrium, we're using hair cells. And while we're at it, we can add those hair cells to our list for the cochlear duct too, right? To the process of hearing. And we could add hair cells to our list for tasting and for smelling. Hair cells are all over the place when we're talking about special senses. So when we're comparing static and dynamic equilibrium, I can't compare them based on the cells they have because their cells are exactly the same. Now, where I find those cells in, that's different between dynamic equilibrium and static equilibrium. When I'm talking about static equilibrium, the location where I find those hair cells is inside something called the macula. Remember, we talked about the macula. That's the part of the saccule and the utricle where I'm actually going to do my detecting of going forward and backwards. The macula is made of something, or well, it's made of hair cells, and on top of those hair cells is something that's kind of like jello called the autolythic membrane. This jello membrane can move back and forth depending on where the otoliths are found. The otoliths. Remember, I told you guys that's my favorite in the static equilibrium process, otoliths. Otoliths are the crystals in your ears, basically, crystals in ear. These little rocks made of calcium carbonate, they move forward or backward depending on what your head's doing. So your detecting of whether you're going forward or backward has to do with these otoliths, these little crystals, rolling forward or backwards. When they roll, the membrane that they're on top of slides, because it's kind of like jello. And when that membrane starts sliding, it pushes on my hair cells. Hey, we should remind ourselves, what kind of gated channel do I find on the hair cells that help me detect forward and backwards, that help me do static equilibrium? Which kind of gated channels are on these guys? Yeah, when we talk about static equilibrium, the kind of channels that we have on our hair cells are mechanically gated. Mechanically gated. When I push them open, remember that they are potassium channels. When I push open that membrane, because there's so much potassium outside of these hair cells, because they're living in endolymph, I push open the channel, in rushes potassium, and that depolarizes my membrane because I rush in that potassium. Mechanically gated potassium channels, that's the way I'm going to detect whether my head's going forward or backwards. Remember that mechanically gated means it's pushed open. So what actually pushes them open are the movement of these otoliths, the movements of these little crystals. When we talk about rotational equilibrium, we're no longer talking about the macula and the otoliths. When I talk about rotational equilibrium, what are some of the words that we would put over here? Let's make our list for rotational equilibrium. Okay, perfect. So my first word, my direct connection, we would say is the crista. If we're trying to figure out where do the hair cells live, in dynamic equilibrium, the hair cells live inside something called the crista. The crista, on top of it, instead of having an autolythic membrane, it has that thing called the cupula. And remember, the cupula is pretty bendy too. It's another kind of jello-like membrane up here. The cupula doesn't slide forward or backwards. The cupula bends. So remember our picture of equilibrium. The cupula is going to bend when your body starts spinning, because that endolymph that's inside the semicircular canals doesn't start spinning at the same time that the rest of your body does. Remember, we talked about inertia, so it's slow to start spinning. When I start spinning, that's going to bend my cupula one direction, and I'll detect that to help me know that I'm rotating. When I stop rotating my body, that fluid keeps spinning for a little while, which is why you feel dizzy, but it's also how I bend my cupula the other direction. That's how I know that I've stopped spinning. So the cupula is the structure that bends to the left or to the right, depending on whether you're spinning or you've stopped spinning. That's what's going to push on the hair cells, or more specifically on the mechanically gated channels that are found on the hair cells. I think that's the big comparisons to make. Let me add one more thing to our list here, because we should be doing this with each of our special senses. And this is reminding ourselves which cranial nerve helps us to detect each special sensation. What cranial nerve helps me with both kinds of balance? What's the number and the name? Yeah, cranial nerve number 8. Cranial nerve number 8. Who's going to be brave and try to spell it? There we go. Vestibulocochlear. Cranial nerve number 8. So CN 8. Vestibulocochlear. Cranial nerve number 8 helps us out with balance. That's the vestibulo part of its name. What else does it help us to do with this cochlear part of its name? Yeah, it also helps us with the process of hearing. Absolutely. So make sure that we know, as we're preparing for exam number 4, make sure that we know that this is my hearing and balance nerve, cranial nerve number 8. When I talk about my vision nerve, what's my vision nerve? You can just give me the number. I won't make you type the other ones. Yep, cranial nerve number 2. That's my vision nerve, the optic nerve. And when we talk about the sense of smell, my cranial nerve for smell, yeah, it's the one in the very front, the olfactory nerve, cranial nerve number 1. And remember that we said there were 2 nerves that were involved primarily in the sense of taste. Their numbers were, yep, 7 and 9. So 7 is the facial nerve. Not vagus, Christina. That's number 10. What's number 9? Oh, I know. Cranial nerve number 9. Anybody remember? That's a terrible one for me to make you type too. Let me put here. Taste is number 7, which we said is facial. Yeah, and it's number 9, which is the glossopharyngeal. Glossopharyngeal nerve. Hey, check this out. Glossopharyngeal means tongue, throat. Remember when we were talking about the papillae on the tongue, how we had the Valate Papillae in the very back, and then we had all of those taste buds that we could see down the throat? Here's where they send their information, in the throat, and the Valate Papillae sending their information here with the glosso part of this nerve. So the tongue-throat nerve, where I've got all of those taste buds in the back of the tongue and down the throat. The facial is going to collect taste information from the more anterior part or the front part of the tongue. Yeah, Christina says she knows it. It's just been a long day. No, I feel that for sure. That's totally fair. It has been a semester, right? Let's be real. It's been quite the semester. Oh my goodness. We're almost done. My daughter has no frame of reference for time, so at least 4 times today she asked me if she can open a Christmas present. It's like, Evelyn, no, it is not. It is still not Christmas. We're 2 hours later. It's still not Christmas. So it's going to be a long 18 days over here. I'm just saying. Yeah, Gloria says she feels that. Yep, that's her too. Okay, so that's the comparison I wanted to make with those types of equilibrium. We've got about 40 minutes left in tonight. Are there any other particular special senses topics that we wanted to talk about tonight, since we're technically wrapping up number 13? While we're thinking, I'll draw you a penguin. 2 penguins in one day, man. You guys are lucky. I am not getting any takers on special senses topics, so I'll open the floor then for spinal cord topics, or basically anything from unit number 4 we want to talk about tonight. And I'll say too, while we are thinking and typing, that there is a review assignment up for unit number 4 to give you a chance to go through and practice. So if you want to figure out what you don't know yet, you can always go back to review the various topics that we had in unit number 4. Okay, I've got a couple of requests for the tracts. Yep, we can talk about the tracts. Gloria's interested in ALS. So fun fact, just this morning I pulled the slides out of, I can store a couple of slideshows in our room here in Blackboard. Just this morning I was like, I'll pull out the slides for lesson number 12. And now we're working on them tonight. So let me import them in again. So the unit number 4 review assignment up. The other thing that's up for you too, and I think I heard a couple of us chatting about this, for the final exam, I have also created for you guys 2 sets of optional review assignments, because it would just be too massive if we put everything in the same assignment. So there's one assignment that helps you remember some of the topics from the first half of the semester, and one half that helps to explain the topics from the second half of the semester. We are going to use our class time on Friday to kind of do a review of the second half of the semester, since I might have some people who won't have taken exam 4 yet on Friday. But do plan to work through that second half of the semester assignment before Friday. Possibly try that first half too, just so that you are as aware of what you need to be studying over the weekend as possible. So those 2 review assignments also available for you. Yeah, Gloria, that will be at our normal time, 9:30. It's just going to be on Friday instead of on Thursday. We'll review for the exam Wednesday morning, and then we'll start reviewing for the final on Friday. Okay, so let me pull up. We're interested. Yeah, so Gloria, no class on Thursday. That's correct. Let me pull up number 12. No, you're totally fine, Gloria. That's what I'm here for. Let's see. Tracts are here. To your question, Nicole, it would be best if you can help me out with topics for Friday. I may try to make a survey like I did for the lab review. So be watching for notes about that. I may make a survey for you in case that'll help me to know how to best prepare. And let me toss this question out too, just because I'm curious. If exam number 4 opened on Wednesday instead of on Thursday, how many of you would take it on Wednesday? Would anybody take it on Wednesday, or would we prefer to take it on Thursday? Because I'm curious if there's any demand for it to open a little bit earlier. A couple of people would. Okay. No, it's totally fine if you would take it on Thursday. I don't care. So I may open it on Wednesday. It doesn't really matter when it opens. It really matters more when it closes. I may open it on Wednesday. I suspect that most people would not take it on Wednesday. But what I'll plan to do is I'll set it tonight to open up on Wednesday. Here's our rule. If you take it on Wednesday, just like if you take it on Thursday or Friday, please don't chat with classmates about what's on it. I know that you all, my friends here, are not going to do that, but my obligatory teacher statement that I have to say, I'll open it a day earlier, which means it'll be open longer. Just make sure we're not chatting about it. Other than that, I have no problem with a few people taking it on Wednesday if you would prefer. That's totally fine. Okay, so let me go up to the question. Okay. So I think the question, it looks like, is getting at when we talk about the types of neurons in a tract. So we didn't review them in generic terms. I just kind of introduced it to us when we were talking about our specific examples. So before we talk more specifically again about the spinothalamic tract, let's talk about these 3 kinds of neurons that we typically find in any spinal cord tract. So the first kind of neuron, or any ascending spinal cord tract, that's an important clarification. In any ascending spinal cord tract, we always have a first order neuron. We always have, I'm going to use orange because yellow is hard to read, second order neuron. And almost always, typically, we're going to have a third order neuron. I mentioned this like 4 weeks ago, when we talked about this together in class. Please never tell me that a first order, second order, or third order neuron are found in a descending tract. This terminology, first order, second order, third order, I only see that when I'm talking about ascending tracts. Hey, if I'm talking about an ascending tract, is that sensory or motor? What are ascending tracts? Yeah, ascending tracts where I send information up, those are my ascending tracts. They're sensory tracts. So first order neurons, when we're thinking about the function or kind of a description of what this kind of neuron is, this is the neuron that detects the sensation. So the neuron that detects, I'll put sensory information. So this is going to be a neuron that starts somewhere in the body. We're going to say starts at a receptor. So for example, in your finger, it starts in your finger and it ends in the dorsal horn of the spinal cord. Spinal cord. We can see that on our picture here. The first order neuron, it's starting somewhere way out here, out in your finger. It comes in through the dorsal root, the thing on the back side, goes into the dorsal horn where it synapses with the second order neuron. Since the second order neuron is found here in the dorsal horn, what would my functional classification for this neuron be? Who lives in the dorsal horn of the spinal cord? Who's back here? I should have checked my timestamps here. So I've got, it looks like I've got a vote or a couple votes, can't tell, for sensory, and I have a vote for an interneuron. This would be a good review for us. When we talk about the kind of neurons that live in the dorsal horn, the kind that have their cell body in the dorsal horn of the spinal cord, which kind of neurons have their cell body back here, their thinking parts back here? Is this a sensory neuron or is this an interneuron that lives in the dorsal horn of the spinal cord? Yeah, so review for us. Remember that in the spinal cord, we've got the 3 gray horns where different neurons live. The neurons that live in the dorsal horn are interneurons. Remember, when I say it lives somewhere, that means its cell body is there. So interneurons live in the dorsal horn. In the lateral horn and in the ventral horn, we have motor neurons that can send out directions. The sensory neuron comes in the dorsal root and brings information there, but the sensory neuron actually lives out on the back side of the spinal cord in that dorsal root. Yeah, in particular, like Nicole mentioned for us, the place where I find the cell body for a sensory neuron is called the dorsal root ganglion. The thing that we talked about a ton in lab, that structure that helps us find the backside. We sometimes abbreviate it DRG, the dorsal root ganglion. The dorsal root ganglion is where sensory neurons live. Interneurons live in the dorsal horn. Motor neurons live in the lateral and the ventral horns. Remember that the motor neurons that live in the lateral horn control smooth muscle, so they're working with organs, and the motor neurons that live in the ventral horn are controlling skeletal muscle, voluntary contractions. So first order neuron is the neuron who lives in the dorsal root ganglion. That's where its cell body is. This is a sensory neuron. It collects sensory information, sends that information into the spinal cord where it synapses with an interneuron. When the interneuron receives that information, remember that we joked, this interneuron says, this is above my pay grade. I can't process this. I've got to send this up. I literally can't even with this. So the interneuron then leaves the gray matter of the spinal cord to enter the white matter of the spinal cord. The white matter of the spinal cord, remember, is where I have a lot of myelin, so I can send messages quickly. My second order neuron starts in the dorsal horn, travels through the tracts, through the white matter of the spinal cord, and ends at the thalamus. It ends at the thalamus. So let's write some notes for ourselves. My second order neuron is an interneuron. It's an interneuron. It starts in the dorsal horn. Dorsal. It travels up the white matter tracts, and it ends in the thalamus. The thalamus. I put a little asterisk there because when we talk about a different tract, we can go somewhere else. So there's my asterisk. Most of the time, we end in the thalamus. The thalamus is where we find our third order neurons. So our third order neurons start in the thalamus, and they end up in the part of the brain where that specific sensory information can be processed. So typically where a third order neuron is going to end up is that thing, remember, that we called the sensory homunculus, the map for where the information goes. Pretty much all of my tracks, that's where we're going, is somewhere different along that sensory homunculus. So let's outline that third order neuron. The third order neuron, I'll put in parentheses, it is also technically an interneuron. It's sending information up to the brain. It starts in the thalamus and it ends in the sensory homunculus, or it ends in the particular part of the brain where I can process that sensory information. So let's look at our descriptions here. First order, second order, and third order neuron. For each of these neurons, make sure you know where they start, make sure you know where they end. In the spinothalamic tract in particular, remember that we talked about how we have more than one that has that name. We have the lateral spinothalamic tract and the anterior spinothalamic tract. Lateral means we're more toward the outside. Anterior means we're more toward the front side. We have two different superhighways, or two different tracks that start in the spinal cord and end up here in the thalamus. If we're sending it through the superhighway on the front, we'd be sending information about the general shape of something. That's what crude touch is, kind of like the shape of something, and pressure, so how much it weighs. If we're sending that information on the lateral side, that information is how hot or cold something is, or that's pain information. I told you this before, and I want to emphasize again, don't memorize the particular types of information that go up these two different tracks. Just know that if I'm going up the anterior tract, I'm a different kind of information than if I'm going up the lateral tract. And if I'm going up a completely different tract, so something that's not spinothalamic, something else, I'm going to have a different kind of information. Each tract in the spinal cord transports different kinds of information. The one other thing I'll mention about the spinothalamic tract is remember that we're always trying to use the name of a tract to figure out if it's ascending or descending. Whenever I see this spino word here, what does spino refer to? Spino? Yeah, spino is the spinal cord. So we're starting in the spinal cord, and thalamic means that the place I end up, or where I'm taking this information to, yeah, is the thalamus. So I start in the spinal cord, I go up toward the thalamus. This is an ascending tract because I start lower than I end up. What questions do we have about the Spinothalamic Tract? I apologize, my daughter's going nuts. Yeah, Gloria, so the lateral horn is actually the one that does smooth muscle. Smooth muscle, we call it the visceral motor neurons, the ones that are in the lateral horn, visceral motor. Somatic motor are the ones that are in the ventral horn. So the question is, the dorsal and lateral horns contain interneurons, and the sensories in the dorsal root ganglion. So the lateral horn contains motor neurons as well, but that's those visceral motor neurons that we were mentioning. So the ones that control smooth muscle in the lateral horn, the ones that control skeletal muscle in the ventral horn, and the interneurons that control either of them would be found in the dorsal horn. So if that's what you were thinking, we're on the same page. We're good. So the other ascending tract we talked about was the spinocerebellar tract. When we talked about the spinocerebellar tract, what was unique about it? What was special about this one? Anybody remember? Yeah, exactly. The spinocerebellar tract is an ascending tract that just has 2 neurons in it. My first order neuron, hey, look at this, that word from today. That first order neuron is a proprioceptor. What did we say that proprioceptors detect again? What are we detecting in this tract? Yeah, this is stretching. Yeah, don't quit, Christina. I'm pretty sure that I told you when we covered this in class to ignore this word because it's a really big word that doesn't actually matter. So we've got some neurons here that are detecting stretching, proprioception. My first order neuron is out in your muscles, and it's detecting how stretched out those muscles are. That information comes in through the dorsal root. There's that dorsal root ganglion. We go into the dorsal horn. When we get into the dorsal horn, we synapse with an interneuron that says, wait a minute, I can't do anything with this. That interneuron, my second order neuron, runs up through the white matter of the spinal cord and goes to the cerebellum. And that's where the spinocerebellar tract ends. If we have 2 neurons, what are we missing? What's the name of the one that we're missing in the spinocerebellar tract? Who's missing? Yeah, the one that we're missing was called the third order neuron. This tract has no third order neuron. And here's where we put the puzzle pieces together. Where did the third order neuron start? Where do I find third order neurons? Yeah, Nicole's right. Third order neurons start in the thalamus. This particular tract, the second order neuron doesn't go all the way up to the thalamus. The thalamus would be up here. The second order neuron goes to the cerebellum instead. It's still an ascending tract because we still go from the spinal cord up to the cerebellum, but I don't have a third order neuron like I normally do. So the spinocerebellar tract is an example of an ascending tract with just 2 neurons in it instead of 3. Normally, what you should know about ascending tracts or sensory tracts is that they have 3 neurons. Here's my exception that proves the rule, if you will, as they like to say. So the spinocerebellar tract just has 2, but the majority of my ascending tracts are going to have 3. How do we feel about ascending tracts? Thumbs up, thumbs down. Christina's doing a handstand. It's that coffee, right? That coffee is is helping you. Ah, yes, and the baby. Yep. Okay, well, let's chat descending tracts then. Hannah's covering her eyes. She can't even. Descending tracts are motor tracts. Motor tracts send information from the brain down to the body. Yeah, so Gloria asked a clarifying question. With an ascending tract, we call it ascending because it's literally sending the information up. Absolutely. This is one of those things where anatomy is actually as simple as it sounds like it is. I promise that it's totally true. An ascending tract, we literally are sending information up. The only kind of information we would send up to the brain from the body is sensory information. The only kind of information we're going to send down from the brain into the body is going to be motor information. So anytime you have a descending tract, we're always sending motor information. The corticospinal tract starts in the cerebral cortex. That's the outside of the cerebrum. And it ends in the spinal cord, or it ends with a neuron in the spinal cord that's going to go out to the body. We start up higher, we end down lower. This is a descending or a motor tract. Whenever we talk about motor tracts, we have 2 neurons in those tracts. We have what's called an upper motor neuron, and we have what's called a lower motor neuron. So let's do what we did before with our first order, second order, third order neuron. Let's talk about where they start and where they end. In a motor tract, the upper motor neuron is going to start in the motor homunculus. Start in the motor homunculus. Remind me in the chat, what's the name of the bump in the brain that has the motor homunculus? Where do I find that motor map? Yeah, so the motor map is in that place called the precentral gyrus. The precentral gyrus. My upper motor neuron, its cell body, its thinking part, is up here in the motor homunculus. Then it's going to travel through the white matter, so travel down through white matter, through another tract, a descending tract. And my upper motor neuron is going to end in the ventral horn of the spinal cord. End in the ventral horn of the spinal cord. When we talked about the ventral horn of the spinal cord, what kind of neuron did we say lived here? Who lives in the ventral horn of the spinal cord? Yeah, Nicole is totally right. When we're in the ventral horn or the lateral horn, we're talking about a motor neuron. In particular, when we are in the ventral horn of the spinal cord, we're doing what is called a somatic motor neuron. So here's my classification for this one. It's a somatic motor neuron. That means it's controlling skeletal muscle. Controlling skeletal muscle. My lower motor neurons go down. They start in the somatic, excuse me, in the ventral horn. So let's write this. They start in ventral horn of spinal cord. They leave through the ventral root, and they end at the skeletal muscle. The lower motor neuron is what actually makes muscle happen, or action happen, what actually causes the muscle to contract. So this synapses with muscles. The upper motor neuron in the brain, I've decided to do my movement. My axon is going to go all the way down through the brainstem, all those parts there, all the way down to the spinal cord, to the ventral horn of the spinal cord. In the ventral horn, it's going to synapse with the lower motor neuron. The lower motor neuron is going to leave the spinal cord and go out to find the muscle we're telling to contract and make the contraction happen. So a motor pathway, a descending pathway, is going to have two neurons every time, upper motor neuron and lower motor neuron. This one should be more obvious, but obviously, please never tell me that there's an upper motor neuron in an ascending tract. Remember, ascending tracts are sensory, so I will never find an upper motor neuron in an ascending tract. I will never find a lower motor neuron in an ascending tract. I only find these in descending tracts. This is an example of, remember, what we called a direct motor pathway, because the directions for these movements come from the cerebrum. They're consciously determined when we talk about the corticospinal tract. Remember that when we talked about the vestibulospinal tract, this was an example of an indirect motor pathway because the directions did not come from the cerebral cortex. Hey, check this out. Vestibulo, that sounds a lot like something we talked about today. What does vestibulo sound like? Yeah, vestibulo, as in the part of the ear that's the vestibule. The vestibule. The vestibule is the saccule, the utricle, and the semicircular canals. The vestibulospinal tract is an indirect motor pathway. This pathway helps me to stay balanced. So it's using the balance information collected by the vestibule of the ear, and it's sending directions based on that. So the vestibulospinal tract is a descending tract. It starts in the brainstem, goes down to the spinal cord. In particular, it's going to help me balance. But again, it's an indirect pathway because we did not start in the cerebral cortex. What questions do we still have about tracts? Are we feeling any better about them? The sideways glance. Oh, I'm sorry, Christina. Christina feels a little bit more confused. Well, if that explanation made it more confusing, yeah, it could be your distraction, or it could just be that we talked about it in a different way. You can always go back and watch the other video where we talked about this. Maybe that'll re-clarify it for you. I'm glad that Mushu made an appearance again, Hannah. I was missing him this morning, man. We were talking about you. We missed you this morning. I'll get out the little lucky cricket. There's the lucky cricket. All right, well, I guess the one other thing we maybe wanted to chat about was ALS. Do we still want to chat about that, or we want to save it? Gloria says yes. Okay, so I will pull that up. We'll chat about that, and then we will call that good for today. I have open office hours. A quick reminder that I have open office hours tomorrow from 9:30 to 11. That's time where I'm not planning to talk about anything. It's unstructured time, so if you have particular questions you want to chat with me about, I will be here. But I'm not gonna like do a class session per se, unless like all of you show up, and then we can talk together, I guess. Our next scheduled class section will be on Wednesday. And on Wednesday, we will go through and talk about any other topics from spinal cord and special senses that we want to review one more time. So we'll do ALS today, and then I'm free tomorrow, and then we'll last class session before the exam on Wednesday. When we talk about ALS, so let me get my typer up here. So ALS is short for amyotrophic lateral sclerosis, ALS, amyotrophic lateral sclerosis. This is essentially a muscle and nerve wasting disease. Patients with ALS will eventually lose control of the muscles in their body. The one that is the most problematic is when their diaphragm, when they lose control of their diaphragm, because then they can no longer breathe. So in normal situations, in you and I, every time our neuron fires, it causes our muscle cells to contract. As our muscle cells contract, that keeps them big and it keeps them strong. Literally the size of the muscle is larger. And the more we use our neurons, the more healthy and the more strong those neurons are as well. They're not wasting away, essentially. In patients who have ALS, the key characteristic of it is muscles that are wasted, or muscles that are much smaller. The word that you'll sometimes see about these muscles, it's called atrophy, when they get smaller. So in ALS, this is an issue of the nervous system talking to the muscles, because when the muscles don't get a message to contract, they start to shrink and waste away. So the issue with ALS is actually a neuron issue. So ALS is a problem with our descending pathways, with our tracts that send information down that go and talk to muscles. So we did an activity in class together. We talked a little bit about this article here. This is, at this point, a really old article. It's like 30 years old now. So this is one of kind of the seminal articles in the field, one of the first articles in studying ALS. The goal of this study was to look at the sizes of the cortical motor neuron and the lower motor neuron. Now, for my friends who were in our session before, we talked about how there's a different name for this cortical motor neuron. What did we call the cortical motor neurons? What was our word for them? Yeah, we called these cortical motor neurons the upper motor neurons. Remember from our discussion of tracts, we said that the upper motor neuron lives in the cerebral cortex. So it's the same thing, it's just a different name for it. So this particular paper, their goal was to look at how the sizes of neurons changed, whether it was the upper motor neuron or the lower motor neuron, leading to ALS, leading to muscle wasting. Essentially, they wanted to figure out, did the brain stop sending messages, or did the neuron that went out to the muscle stop sending messages? So they took some biopsies, some brain biopsies. They looked at cells that were in the foot and the tongue areas of the precentral gyrus. So they looked at that map, that motor homunculus. They took pieces of the brain tissue that were supposed to control the foot and that were supposed to control the tongue, and they looked at the size of the cells there. And then they went down and they looked at the size of the neurons that lived in what's called the hypoglossal nucleus, which is the part of the brain stem that controls the tongue, and down in the ventral horns of the lower spinal cord, where we're sending out neurons that control the foot. They looked at the upper motor neuron up here in the precentral gyrus. They looked at the lower motor neurons in these two places. What they found in this study is that both the upper motor neurons, the cortical neurons, and the lower motor neurons were both smaller in patients who had ALS. So we basically can't tell if it's a chicken or an egg thing. Did the upper motor neuron stop talking, so the lower motor neuron got smaller? Or did the lower motor neuron stop listening, and that's why the cortical neuron stopped talking? We didn't really find out what caused these neurons to get smaller, but we did find out that both of the neurons in a descending pathway are smaller. Both of them have atrophied, meaning that both of them are no longer sending messages for the skeletal muscle to contract. What questions do we still have about ALS? Jacqueline's speaking to me in wingdings. Ah, got it. Makes a little bit more sense. Little one at the computer. Yeah, go for it, Gloria.
Okay, so just to clarify, because every time I took the test, I kept getting it wrong, and now I'm starting to realize it's because the motor neurons don't have anything to do with the first, second, and third order neurons, because that has to do with the sensory part. Is that right?
Exactly. Yep. I only see first, second, and third order in sensory pathways. Yeah, absolutely.
Okay. Perfect. Thank you so much.
You're welcome. I'm glad it was a simple thing like that. Those are frustrating, but they're also good to have a simple error to correct. All right. Well, that's all I've got for today. Like I mentioned, I will be here tomorrow in the room just chilling out from 9:30 to 11. So plan to stop by if you have questions. Otherwise, I hope to see a lot of you guys on Wednesday at 9:30. We will review for the exam. And like I mentioned, I'll go ahead and set that exam up. So if you want to take it on Wednesday instead of Thursday, I don't care. Get it done. So I'll set that up so that is available for you. So have a great evening. I will stick around in the chat for any last minute questions that we have.